Your endomembrane system. That's going to include your nuclear envelope, your lysosomes, your vesicles, your rough ER, your smooth ER, your Golgi apparatus, and the very outer membrane. From your nuclear envelope to your cellular membrane is your endomembrane system. It's just an interconnected organelle system involved with bringing stuff out of the cell. You could say bringing stuff out of the cell or bringing stuff into the cell. It's an interconnected network when you look at the grander scheme of a cell and all of its parts. Let's start off with the rough ER. The rough ER is going to usually be contiguous with the nuclear envelope, and it's going to be rough because it has ribosomes attached to it. And then smooth ER, that's actually going to be a ribosome-free region of your endoplasmic reticulum. So no beads of rough or no beads of ribosomes around the smooth ER. We know that our rough ER, that's going to be a great construction site for new proteins. It's going to have chaperone proteins, proteins involved with protein folding, and then also, again, it's beaded with ribosomes that will squeeze chains of amino acids into that rough ER space where the proteins will fold the way they're supposed to be folded. So your rough ER has ribosomes. It's, again, that's going to be the construction site for your proteins. Your ribosomes are just going to be the construction workers, if you think about it that way. Your ribosomes are the sites for, or the workers of protein synthesis. They're going to be the structures that help you translate mRNA into sequences of amino acids we would call proteins. All right, so in eukaryotes, just to go on a little bit of a tangent, your ribosomes could be found in the rough ER, they could be found in the cytoplasm, they could be found within mitochondria. Mitochondria are kind of like their own organism, and they're, again, bound to the rough ER, and then you're also going to find them in chloroplasts, kind of like their own organism. So they consist of a type of RNA called rRNA, or ribosomal RNA, and more than 50 other proteins. Your smooth ER, that's the site where you're going to typically synthesize lipids. It's also a major site where you process toxins and drugs, so very commonly associated with detox. And so therefore it's found, your smooth ER is going to be found abundantly in your liver cells, which we call hepatocytes. So all of your liver cells are going to have a large abundance of smooth ER. And it's also a place where you would store and release calcium ions in some cells, like neurons, for example, which calcium release is involved in neurotransmission. Okay, so Golgi apparatus. That's going to be, if you remember from previous biology courses, kind of like the post office of your cell. So it's a flattened membranous, or it's going to consist of flattened membranous sacs and small membrane enclosed vesicles. It's pretty much where you receive your rough ER products and you repackage them to send to wherever they need to go within the cell. So your Golgi apparatus has three roles. It's to modify endoplasmic reticulum proteins. It's going to sort and ship cell products. And then it's also going to make some cell wall carbs for signaling molecules, for example. So here you have your rough endoplasmic reticulum. It should be beaded with ribosomes. You're going to synthesize proteins, send them out in vesicles to your Golgi apparatus. And then within your Golgi apparatus, you might refold those proteins. You might tag those vesicles containing those proteins with signals to direct the vesicle to where it needs to go within the cell, such as directly outside of the cell to the extracellular space. So in the absence of specific targeting signals on these vesicles, proteins are carried to the membrane by default. We call this constitutive secretion. So in this case, constituted secretion is just by default tagged to be released outside of the cell. Now, you could simply not tag it to be sent outside of the cell, Proteins can be diverted from the constitutive pathway and targeted for other destinations, such as lysosomes. So you could embed a different signal into that vesicle to direct it elsewhere within the cell. So by default, we send it outside of the cell unless told otherwise. Your lysosomes, those are going to be vesicles containing digestive enzymes. And the function of your lysosomes is, it's kind of like the garbage disposal, or you could say like the recycling area, the recycling organelle. They're going to be these sacs of a bunch of enzymes that are hydrolytic, or they will break down larger molecules into smaller molecules. So sites for breakdown or lysis of food as well as foreign material, or it could even break down old, used, misfolded proteins or organelles that are dysfunctional. So they could be sites where digestion of spent cellular components occurs. And we call this process autophagy. Autophagy can be broken down. Phagy means to eat, and then auto means self, so self-eat. So in this case, it just shows a few different pathways. You can send, through pathway 1, you could send products to be excreted from the cell through exocytosis. You can create vesicles containing proteins that could serve as cell signals on the outside of the cells, embedded into the membrane. And in this case, the third pathway that they show is that you could also create these things called lysosomes containing acid, hydrolase enzymes. You could just say enzymes that would break down a multitude of different molecules down into their base components. And so in this case, we have what we would call a phagosome. So phago meaning to eat, zome meaning it's a vesicle, some kind of vesicle, so a package. So this is going to be, a phagosome maybe from receiving stuff from the outside of the cell. So through endocytosis, maybe we've collected some extracellular components, maybe some kind of foreign material, or it could just be a phagosome composed or containing dysfunctional cellular parts. All right, next we have mitochondria. We know that that's the powerhouse of the cell. And the role that mitochondria play is they convert potential chemical energy of food, or you could say fuel, into a form that we can use within the cells. So they take potential energy from sugar, for example, and they convert it into a usable type of energy called ATP. And they do all of that through what we would call aerobic cellular respiration. Aerobic meaning with the use of oxygen, and it's a way of breaking down foods completely. So there's some components of the mitochondria. We have a double membrane. So it's got an outer membrane as well as an inner membrane. And in that inner membrane, we have all of these folds. We call those folds cristae. So the reason why we have all of these folds in the inner membrane, or all of these cristae within the inner membrane, is because it increases surface area. And the reason why we want more surface area in this inner membrane is because it provides more space to embed enzymes or proteins involved in the electron transport chain. So the more folds we have, the more proteins we can embed into that membrane. And so if we want to be a very efficient mitochondria, we're going to have a lot of folds within our inner membrane so we can fit more enzymes and proteins. And then lastly, we have the mitochondrial matrix. So the mitochondrial matrix is just going to be the pulpy juice within the that inner membrane of the mitochondria. And that's going to contain all of the enzymes associated with pyruvate oxidation or the citric acid cycle. Now we have plastids. These are just going to be organelles that we find in plants and some protists. Protists are going to be a type of single-celled eukaryotic cell, or single-celled eukaryotic organism. Now plastids can have a variety of functions. A few of those subtypes of plastids, we have chromoplasts, which are just going to be plastids that contain a bunch of pigments. You could also have amyloplasts, which is another type. That's going to be a type of plastid that stores starch. And then you also have chloroplasts. As we know, chloroplasts are going to be kind of like that site for photosynthesis. And then just to dive a little bit into chloroplasts. So chloroplasts, we know those are going to be the sites where photosynthesis occurs, and your chloroplasts are going to have it's going to be doubly membraned, kind of like our mitochondria. So chloroplasts are surrounded by two layers. So you have an outer membrane as well as an inner membrane within your chloroplasts. And within those chloroplasts, you have these things called thylakoids, which are these flat membranous sacs where your light reaction of photosynthesis is going to occur. A group of those stacks, or a stack of stacks, we would call a grana, and a multiple stacks we would call granum. So that granum is the plural form of grana. And then stroma, that's going to be the fluid that suspends all of this stuff on the within that inner membrane, and that's also where the Calvin cycle occurs, or the light-independent reaction. Now we have peroxisomes. So these are going to be small organelles that compartmentalize toxic peroxides and break them down, or you could say neutralize them down into water and oxygen. Your mitochondria are going to be a prime source of peroxide production that usually results from the incomplete reduction of oxygen during the last step of the electron transport chain. So you'll create peroxides as a result of, improper reduction within your mitochondria, and then we have another organelle that functions to neutralize the harmful peroxides. And then we have vacuoles. So you're going to find these things in plants, you can find them in fungi, you can find them in protists, and they're typically filled with an aqueous solution. Now they can have multiple roles. So your vacuoles can store wastes and toxins. They're going to be responsible for the turgor pressure within plant cells, for example. So if you have like a nice taut or crispy lettuce, it's going to have a lot of turgor or pressure within their central vacuoles of those cells. You can also store pigment in there. You could also store toxins. If you're trying to ward off herbivores, if you're a plant that's trying to ward off herbivores, it could also be a site for digestion. And it's also an organelle that can be used for movement in contractile vacuoles, for example. So let's take a look at some contractile vacuoles. As you'll notice, the contractile vacuoles will take up fluid and expel that fluid in order to move the organism. So here we're going to zoom in on a nice contractile vacuole. You see the contractile vacuole? It's very nice. Contractile vacuole.